Sholihah.
Kita perlu mensuplai berbagai nutrisi dari berbagai makanan. Tubuh kita tidak dapat menggunakan makanan seperti itu pada sistem pencernaan. Proses pencernaan kimia menggunakan protein dan enzim yang berbeda untuk memecah partikel makanan menjadi nutrien yang digunakan sel-sel untuk menyerap. Sintesis protein membutuhkan bahan dasar asam amino dan berlangsung di dalam inti sel dan ribosom. Untaian panjang tersebut adalah DNA. Gen adalah potongan untai rangkap DNA yang tersusun dari polimer nukleotida. Wilayah ini dimulai dari wilayah promotor dan berakhir di wilayah terminator. Untuk beberapa gen, RNA dikodekan untuk mensintesis protein. Secara garis besar, sintesis protein dibagi menjadi dua proses, yaitu transkripsi dan translasi. Dalam sel eukariotik, transkripsi terjadi di inti sel di mana DNA digunakan sebagai tempat untuk membuat RNA duta. Sedangkan translasi terjadi dalam sitoplasma. Informasi yang terkandung dalam RNA duta digunakan untuk membuat polipeptida. Selama transkripsi, RNA pada gen digunakan sebagai tempat untuk membuat untai RNA duta dengan bantuan RNA enzim polimer. Proses transkripsi melalui 3 tahap, yaitu initiation, elongasi, dan terminasi. Pada proses initiation, wilayah promotor berfungsi sebagai tempat RNA polimer melekat pada molekul DNA sehingga menyebabkan sebagian dari double helix terbuka. Akibat terbukanya pita DNA, basa-basa pada salah satu pita menjadi bebas, sehingga memberi kesempatan basa-basa pasangannya menyusun RNA duta. Proses ini disebut elongasi. Pada proses terminasi, RNA polimer menuju bagian terminator gen. Setelah transkrip RNA duta selesai dicetak, untai DNA dan transkrip RNA memisahkan antara satu sama lain. RNA duta yang membawa kode untuk protein disebut ekson, dan bagian yang tidak membawa kode disebut intron. Intron splicing adalah proses penambahan 5' concat dan 3' polyacyl, sedangkan spliceosome adalah proses menghilangkan segmen intron dan bergabung dengan ekson yang berdekatan untuk menghasilkan RNA untai matang yang kemudian meninggalkan inti sel menuju sitoplasma. Basa nitrogen dikelompokkan menjadi 3 kode huruf. Kode ini disebut kodon. Kode genetik meliputi 64 kodon. Ada 4 kodon khusus, yaitu pada kodon start untuk memulai dan 3 kodon stop untuk berhenti. Translasi dimulai dengan RNA duta untai mengikat subunit ribosom kecil pada kodon start. Asam amino dibawa ke ribosom oleh RNA transfer. Terjadi pasangan antara kodon RNA duta dan antikodon dari RNA transfer. Setelah molekul RNA transfer inisiator mengikat kodon start, subunit ribosom besar mengikat. Dan tahap inisiasi selesai. Dalam subunit ribosom besar terdapat 3 daerah yang berbeda, yang disebut daerah E, P, dan A. Pada tahap elongasi, molekul RNA transfer melalui pasang basa komplementer dari kodon dan antikodon asam amino dibawa ke untai RNA duta. Molekul RNA transfer memuat sesuai dengan asam amino tertentu. Molekul RNA transfer dibebankan mengikat ke situs A dan mendorong satu kodon ke kanan, di mana molekul RNA transfer sekarang bermuatan keluar dari situs E, dan situs A ini terbuka untuk menerima molekul RNA transfer berikutnya. Elongasi akan berlanjut sampai kodon stop tercapai. Faktor pelepas melekat pada situs A pada kodon stop. Polipeptida dilepaskan dari RNA transfer di sisi P, kemudian subunit ribosom terlepas. Tujuan translasi adalah untuk menghasilkan polipeptida cepat dan akurat. Setelah pemisahan, polipeptida menjadi protein. Agar enzim pencernaan disekresikan ke dalam perut atau usus, polipeptida diterjemahkan ke dalam retikulum endoplasma, diubah saat melewati Golgi. Kemudian disekresikan menggunakan vesikel melalui membran plasma dari sel ke dalam lumen saluran pencernaan. Protein yang diperlukan bagi sebagian besar tubuh diproduksi melalui proses transkripsi dan proses translasi.
what this is? It is the longest word in the world. Like anywhere, any language, more than 189,000 letters. If you were to write it down, though I don't know why you would, it would fill up more than 100 pages, and if you could actually say it without, like, breaking your face, it'd take about 5 hours. What the frick is this word? Well, it is the name of the longest known protein on Earth, and it's actually in you right now. Because of its enormous size, it was given the name Titan by scientists, and that's with 2 Y's. And it's protein that helps give some like the springiness to your muscles. Today we're going to be talking about DNA and how it, along with 3 versions of its cousin RNA, unleash chemical kung fu to synthesize proteins just like this. So this is going to take a while to explain. So how about we make ourselves some Hot Pockets? Those are my favorite. Ham and cheese. Every time I take a bite, I wonder, how do they do it? How do they pack exactly the same flavor into every foil cardboard wrapped food-ish item? Clearly there's got to be some super secret instruction manual kept and a location known only to 2 people. And since I'm talking about biology here, that brings up a related question. How did I get built from the DNA instructions and biological molecules we've been talking about? Today, that's what I'm going to do. Not actually make Hot Pockets or a person, but I'm going to be talking about DNA transcription and translation, which is how we get made into the delicious things that we are today, though hopefully none of us know how delicious people are. Animals, plants, and also Hot Pockets really are nothing more than salty water, carbohydrates, fats, and, proteins combined in precise proportions following very explicit instructions. Let's say I want to make my own Hot Pocket. I would have to, 1, break into the lair of the Hot Pocket company holding the secret manual. 2, read the instructions on how to make the machinery to produce the Hot Pocket and the proportions of the ingredients. 3, quickly write down that information in shorthand before I get caught by the Hot Pocket police. 4, go home, follow the instructions, build the machinery, and mix the ingredients together until I have a perfect Hot Pocket. And that is how we get us. Very simply, inside the cell's nucleus, the DNA instruction manual was copied gene by transcription onto a kind of RNA and then taken out of the layer where the instructions are followed by the process of translation to assemble amino acid strings into polypeptides or proteins that make up all kinds of stuff from this titan down here to the keratin in my hair. But most of the polypeptides that get made aren't structural proteins like hair, they're enzymes which go on to act like the assembly machinery, breaking down and building and combining carbohydrates and lipids and proteins that make up variations of cell material. So enzymes are just like whatever ingenious machinery they use the factory to make this. Okay, let's start out in the layer. The nucleus. The length of DNA that we're going to be transcribing onto an RNA molecule is called our transcription unit. And let's say, in today's example, that it's going to include the gene that transcribes for our friend Titan, which in humans, at least, occurs on chromosome 2. Now, each transcription unit has a sequence just above it on the strand, and that's called upstream. Biologists call that upstream on the strand, and that sequence sort of defines when the transcription unit is going to begin. This special sequence is the promoter, and it almost always contains a sequence of 2 of the 4 nitrogenous bases that we talked about in our last episode: adenine, thymine, cytosine, and guanine. Specifically, the promoter is a really simple repetition. We got thymine, adenine, thymine, adenine, and then A, and then on the other side, AT. Because you know how this works, right? This is called the ta-ta box. It's nearly universal, and it helps our enzyme figure out where to bind to the strand. Now you'll remember from our episode about DNA structure that DNA strands run in one of 2 directions, depending on which end of the strand is free and which end has a phosphate bond. One direction is 5' to 3', and the other direction is 3' to 5'. In this case, upstream means toward the 3' end, and downstream means toward the 5' end. So the first enzyme in this process is RNA polymerase, and it copies the DNA sequence downstream of the ta-ta box. That's towards the 5' end, and copies it into a similar type of language, messenger RNA. Quick aside, so you'll notice that to read the DNA in order to make enzymes, we need an enzyme in the first place. So it kind of gets chicken egg here. We need the enzyme to make the DNA and the DNA to make the enzyme. So where did RNA polymerase come from in the first place if we haven't made it yet? What an excellent question. It turns out that all of these basic necessities get handed down from your mom. She packed quite a lot more into her egg than just her DNA, so, we had a healthy start. So, thanks, Mom. So the RNA polymerase binds to the DNA at that TATA box and begins to unzip the double helix. Working along the DNA chain, the enzyme reads the nitrogenous bases, those are the letters, and helps the RNA version of those nitrogenous bases floating around in the nucleus to find their match. Now, you might also recall from our previous episodes that nitrogenous bases only have one counterpart that they can bond with. But RNA, which is the pink one here, doesn't have thymine like DNA does, which is the green and the blue. Instead, it has uracil. So U appears here in T's place as a partner to adenine. As it moves, the RNA polymerase rezips the DNA behind it and lets our new strand of messenger RNA peel away. Eventually, the RNA polymerase reaches another sequence downstream called a termination signal that triggers it to pull off. Now, some finishing touches before this info can safely leave the layer. First, a special type of guanine is added to the 5' end. That's the first part of the mRNA that we copied, and that's called the 5' cap. On the other end, it looks like I fell asleep with my finger on the A key of my keyboard, but another enzyme added about 250 adenines onto the 3' end. This is called our poly-A tail. These caps on either end of the mRNA package make it easier for the mRNA to leave the nucleus. They also help protect it from degradations from nearby passing enzymes, while also making it easier to connect with other organelles later on. But that's still not the end of it. As if to try to confuse me to protect the secret Hot Pocket recipe, the original recipe book also contains lots of extra misleading information. So just before leaving the nucleus, that extra information gets cut out of the RNA in a process called RNA splicing. And it's something like editing this video. This process is really complicated, but I just had to tell you about two of the key players because they have such cool names. One, the SNRPs, which are small nuclear ribonucleoproteins. These are a combination of RNA and proteins, and they recognize the sequences that signal the start and end of the areas to be spliced. SNRPs bunch together with a bunch of other proteins to form the spliceosome, which is what does the actual editing, as it were, breaking the junk segments down so their nitrogenous bases can be reused in DNA or RNA, and sticking together the two ends of the good stuff. That good stuff that gets spliced together, by the way, are called the exons because they will eventually be expressed. The junk that gets cut out are just the intervening segments or the introns. The material in the introns will stay in the nucleus and get recycled. So, for instance, Titan down there is thought to have hundreds of exons when it's all said and done, probably more than 360, which may be more than any other protein. And it also contains the longest intron in humans, some 17,000 base pairs long. Man, Titan, it is just a world record holder. So now that it's been protected and refined, the messenger RNA can now move out of the nucleus. Okay, so a quick review of our Hot Pocket Mission Impossible caper so far. We broke into the layer containing the instructions, we copied down those instructions in shorthand, we added some protective coatings, and then we cut out some extra notes that we didn't need, and then we escaped back out of the layer. Now I have to actually read the notes, make the machinery, and assemble the ingredients. This process is called translation. So next, rewind your memory or just watch that video again to the episode about animal cells. Do you remember the rough endoplasmic reticulum? I hope you do. Those little dots on the membranes are the ribosomes. And the processed messenger RNA gets fed into a ribosome like a dollar bill into a vending machine. Ribosomes are a mixture of protein and a second kind of RNA called ribosomal RNA, or rRNA. And they act together as a sort of workspace. rRNA doesn't contribute any genetic information to the process. Instead, it has binding sites that allow the incoming mRNA to interact with another special type of RNA, the third in this caper, called transfer RNA, or tRNA. And tRNA really might as well be called translation RNA because that's what it does. It translates from the language of nucleotides into the language of amino acids and proteins. On one end of the tRNA is an amino acid. On the other end is a specific sequence of 3 nitrogenous bases. These two ends are kind of matched to each other. Each of the 20 amino acids that we have in our body has its own sequence at the end. So if the tRNA has the amino acid methionine on one end, for instance, it can have UAC as the nucleotide sequence on the other end. Now it's just like building a puzzle. The mRNA slides through the ribosome reads the mRNA 3 letters at a time, each set called a triplet codon. The ribosome then finds the matching piece of the puzzle, a tRNA with 3 bases that will pair with the codon sequence. That end of the tRNA, by the way, is called the anticodon. Sorry for all the terminology. You need to know it! And of course, by bringing in the matching tRNA, the ribosome is also bringing in whatever amino acid is on that tRNA. Okay, so starting at the 5' end of the mRNA that's fed into the ribosome, after the 5' cap, for almost every gene, you find the nucleotide sequence AUG on the mRNA. The ribosome finds a tRNA with the anticodon UAC, and on the other end of that tRNA is methionine. The mRNA, like a mile-long dollar bill, keeps sliding into the ribosome so that the next codon can be read, and another tRNA molecule with the right anticodon binds on. If the codon is UUA, then the matching tRNA has an AAU on one end and a leucine on the other. And if the mRNA has an AGA, then the matching tRNA has a UCU on one end and an arginine on the other. In each case, that new amino acid gets connected onto the previous amino acid, starting a polypeptide chain, which is the very beginning of a protein. But it turns out that there are lots of different ways to read this code, because UUA is not the only triplet that codes for leucine. UUG does too, and arginine is coded for by 6 different triplets. This is actually a good thing. It means we can make a few errors in copying and transcribing and translating DNA, and we won't necessarily change the end product. The process continues with the mRNA sliding in a bit more, and the ribosome bringing in another tRNA with another amino acid, and that amino acid binding to the existing chain, and on and on and on and on and on. Sometimes for thousands of amino acids to make a single polypeptide chain, for example. This whole word is basically just the names of the amino acids in the sequence in the order in which they occur in the protein, all 34,350 of them. But before we can make our own Hot Pockets, and that string of amino acids becomes my muscle tissue, we have some folding to do. That's because proteins, in addition to being hella big, can also contort into very complex and downright lovely formations. One key to understanding how a protein works is to understand how it folds. And scientists have been working for decades on computer programs to try and figure out protein folding. Now, the actual sequence of amino acids in a polypeptide, what you see scrolling along down there, is called its primary structure. One amino acid covalently bonded to another, and that one to another, and to another in a single file. But some amino acids don't like to just hold hands with 2 others. They're a bit more promiscuous than that. The hydrogens on the main backbone of the amino acids like to sometimes form bonds on the side, hydrogen bonds, to the oxygens on amino acids a few doors down. When they do that, depending on the primary structure, they bend and fold and twist into a chain of spirals called a helix. We sometimes also find several kink strands laying parallel to one another called pleated sheets. All those hydrogen bonds and pleated sheets are what makes silk strong, for instance. So in the end, our promiscuous amino acids lead to wrinkled sheets. These hydrogen bonds are what help give these polypeptides their secondary structure. But it doesn't end there. Remember the R groups that define each amino acid? Well, some of them are hydrophobic. And since the protein is in the cell, which is mostly water, all of those hydrophobic groups try to hide from the water by huddling together. And that can bend up the chain some more. Other R groups are hydrophilic, which, if nothing else, means that they like to form hydrogen bonds with other hydrophilic R groups. So we get more bonding and more bending, and our single file line has now taken on a massively complex 3-dimensional shape. It also explains why I can fix my bed head by wetting my hair with water. The water helps break some of those hydrogen bonds in the keratin, which relaxes its structure. That way I can comb it out, and when it dries, those bonds reform, and voila, perfect hair. All of this shape caused by bonding between R groups gives our polypeptide its tertiary structure. So now we have a massively contorted polypeptide chain, and it actually contorts very precisely. Sometimes just one chain is what makes up the whole enzyme or protein. In other proteins, like hemoglobin, several different chains come together to form a quaternary structure. So quick review of structure. The sequence is the primary structure. The backbone of hydrogen bonds forming sheets and spirals are the secondary structure. R group bonds are tertiary, and the arrangement of multiple proteins together give the quaternary structure. These polypeptides are either structural proteins, like this thing at the bottom here, that you can find in your muscle or in my Hot Pocket. They might also be enzymes. Enzymes like do stuff. They can cut up biological molecules like I do with the chef's knife. They can mix stuff, and they can put stuff together. So from that one recipe book, we get all the ingredients and all the tools necessary to make me, which is better than a Hot Pocket. Would you all agree? Now take your time with this stuff. Feel free to watch the episode a couple of times, because next week we're going to talk about how cells swap all of this genetic information through reproduction. Thank you for watching this episode. By now, you should probably know how this works. You can click on any of the links over there, and it'll take you back to that point in the show, as long as you're not watching on your cell phone. It doesn't work on.
Halo adik-adik, jumpa lagi bersama kakak dalam pembahasan soal ujian nasional biologi SMA. Adik-adik, untuk soal kali ini, kita akan membahas tentang soal yang terkait dengan materi sintesa protein. Adapun soalnya adalah sebagai berikut. Di bawah ini merupakan tahap-tahap sintesis protein. Yang pertama, mRNA meninggalkan DNA menuju ke ribosom. Selanjutnya, DNA melakukan transkripsi sehingga terbentuk mRNA. Yang ketiga, asam amino berderet sesuai dengan kode mRNA. Yang keempat, tRNA menerjemahkan kodon yang dibawa mRNA. Yang kelima, protein yang terbentuk merupakan enzim yang mengatur metabolisme sel. Dan yang terakhir, yang keenam, tRNA mencari dan membawa asam amino yang sesuai dengan kodon yang dibawa tRNA. Pertanyaannya, urutan yang benar dari proses sintesa protein tersebut adalah? Adik-adik, untuk menjawab soal ini, Kakak akan menjelaskan sekilas tentang sintesa protein. Adik-adik, sintesa protein ini mengalami dua proses. Yang pertama dinamakan dengan transkripsi. Selanjutnya, yang kedua dinamakan dengan translasi. Kedua proses ini terjadi pada tempat yang berbeda. Transkripsi ini terjadi pada nukleus atau inti sel. Sedangkan translasi terjadi pada ribosom. Oke adik-adik, kita akan mulai dari transkripsi. Transkripsi merupakan proses pembentukan mRNA dari DNA sense. Jadi di sini kakak akan tulis DNA sense akan membentuk mRNA. Inilah yang dinamakan dengan transkripsi yang terjadi di nukleus. Selanjutnya, berdasarkan mRNA yang telah diperoleh dari DNA sense, akan dilakukan proses yang dinamakan dengan translasi. Di mana translasi ini adalah yang pertama, ada tRNA yang akan mengikat asam amino yang sesuai dengan mRNA. Jadi adik-adik sekali lagi, tRNA di sini akan mengikat asam amino yang sesuai dengan mRNA. Selanjutnya, nanti setelah tRNA membentuk asam amino yang sesuai dengan mRNA, asam amino ini akan berikatan seperti ini. Asam amino yang terbentuk jumlahnya maupun jenisnya tergantung dari mRNA. Asam amino ini nantinya akan bergabung membentuk protein. Ya, demikian adik-adik penjelasan sekilas tentang sintesa protein. Dari penjelasan kakak tadi, mari kita perhatikan soal yang telah tersedia. Jadi, hal yang perlu kita cari pertama kali adalah untuk mengurutkan sintesa protein, mari kita cari pernyataan yang mencerminkan proses pertama itu adalah transkripsi. Di mana pada tahapan-tahapan ini yang memperlihatkan proses transkripsi adalah nomor 2. DNA melakukan transkripsi sehingga terbentuk mRNA. Jadi nomor 2 ini adalah urutan pertama. Selanjutnya, setelah terbentuk mRNA, akan dilanjutkan dengan mRNA meninggalkan DNA menuju ke ribosom. Jadi nomor 1 ini adalah urutan kedua. Setelah mRNA menuju ke ribosom, selanjutnya tRNA menerjemahkan kodon yang dibawa mRNA. Nomor 4 adalah urutan ketiga. Selanjutnya, setelah tRNA menerjemahkan kodon yang dibawa mRNA, tRNA mencari dan membawa asam amino yang sesuai dengan kodon yang dibawa tRNA. Jadi nomor 6 ini urutan keempat. Selanjutnya, asam amino berderet sesuai dengan kode mRNA. Jadi urutan ketiga ini, tahapan ketiga ini adalah urutan kelima. Selanjutnya yang terakhir, protein yang terbentuk merupakan enzim yang mengatur metabolisme. Jadi produk akhirnya di sini adalah protein. Jadi protein ini adalah urutan terakhir, yaitu urutan ke-6. Oke adik-adik, urutan yang benar dari proses sintesa protein tersebut adalah, yang sesuai adalah option nomor C, yaitu tahapan dimulai dari tahapan nomor 2, nomor 1, nomor 4, nomor 6, nomor 5, dan diakhiri dengan tahapan nomor 3. Jadi jawabannya adalah C.
Halo adik-adik, jumpa lagi dengan kakak dalam pembahasan soal-soal ujian nasional Biologi SMA. Adik-adik, pada pembahasan soal kali ini kita akan membahas tentang soal yang terkait dengan sintesis protein. Oke adik-adik, adapun soalnya adalah sebagai berikut. Sintesis protein disusun oleh 2 tahapan, yakni transkripsi dan translasi. Keduanya memiliki mekanisme sebagai berikut. Nah, di sini adik-adik sudah diberikan beberapa pernyataan, ya, di mana pernyataan ini secara tidak langsung menjelaskan tentang perbedaan antara transkripsi dan translasi. Nah, adik-adik, sekarang berarti kita akan membahas tentang sintesis protein. Nah, adik-adik, sintesis protein di sini terdiri dari 2 tahapan, ya. Yang pertama itu adalah tahapan yang dinamakan dengan transkripsi. Selanjutnya, yang kedua itu adalah tahapan yang dinamakan dengan translasi. Nah, adik-adik, adapun transkripsi di sini merupakan tahapan yang ditandai dengan pembuatan RNAD atau kodon atau mRNA dari DNA sense. Jadi di sini nanti DNA sense ya akan diubah menjadi mRNA atau RNAD atau mungkin kodon. Selanjutnya, adik-adik ya, di sini ada translasi. Translasi ini adalah ya, yang pertama pengikatan tRNA, oke, dengan asam amino. Jadi di sini asam amino, adik-adik. Jadi terikat dengan asam amino. Asam aminonya ini yang diikat oleh tRNA sesuai dengan kodon, adik-adik. Selanjutnya nanti translasi ini di sini nanti pembentukan protein atau polipeptida. Jadi setelah asam amino terbentuk, adik-adik sesuai dengan pesanan kodon, jadi nanti asam amino ini akan saling berikatan sehingga terbentuk yang namanya polipeptida atau protein. Jadi di sini pembentukan polipeptida atau protein, adik-adik. Seperti ini. Jadi dari penjelasan kakak tadi tentu kita dapat menjawab pernyataan ini, adik-adik. Jadi dari pilihan yang diberikan pada soal ini, yang paling tepat itu adalah
yang C, adik-adik, di mana transkripsi di sini merupakan proses mencetak RNAD atau mRNA atau kodon. Selanjutnya, kalau translasi berarti proses menerjemahkan urutan basa molekul RNAD ke dalam urutan asam amino polipeptida. Karena seperti yang kakak katakan tadi, asam amino yang diikat oleh tRNA akan sesuai dengan pesanan RNAD. Seperti itu. Jadi benar. Ya adik-adik, jadi proses, sekali lagi translasi, proses menerjemahkan urutan basa molekul RNAD ke dalam urutan asam amino polipeptida. Oke adik-adik, demikian penjelasan kakak tentang sintesis protein, yang secara tidak langsung membedakan antara tahapan transkripsi dan translasi, di mana kedua-duanya merupakan tahapan pada sintesis protein. Demikian.